All right. So this is going to be the second installment of general and special senses. Last time we talked about general senses. Remember that in general senses, the receptors are spread throughout the body. And that is temperature, pain, touch, pressure, vibration, and proprioception or body position. I guess specifically the position of joints. We're going to be looking at your specialized senses, which are your concentrated receptors inside of an organ. And that'll be olfaction, which is smell, taste, which is gustation, vision, balance, and hearing. Let's go ahead and start with your specialized senses here. Smell, otherwise known as olfaction, allows you to use specialized chemoreceptors to detect different molecules within the air. Your organ of smell is not your nose, which is just the part that sticks out of your face, but rather it is what's called the olfactory bulb, which is a bundle of nerves up here at the base of the cranial cavity that allows certain nerves to pass through the bone that separates your cranial cavity from your nasal cavity. Now, this is really too big. This diagram, it shouldn't be that big, but it gives you an idea of approximately where it is located. And so that passes down into the top of the nasal cavity. This piece of bone here that separates the nasal cavity from the cranial cavity is called the cribriform plate, and that separates the two. In very rare occurrences, if there's some sort of problem here, sometimes spinal fluid or cerebrospinal fluid can leak down into a person's nose, giving them a perennially runny nose. But that's in the case of some sort of major injury, something's wrong. Otherwise, nothing passes through this other than these nerves. So these nerves are located at the top of the nasal cavity, and approximately 10 to 20 million receptor cells in an area that's 5 centimeters square. That's approximately the size of a postage stamp. And so when you inhale, your nose, which this gives the appearance that your nose is just like totally empty. It's not. It's got folds in it, which make the air spin around as it goes through. Any sort of molecules that are carried on that will spin around and connect to these or touch these chemoreceptors. And if the right chemoreceptor sites are stimulated, that will then send a signal that you are smelling this particular smell. Now, we tend to think because dogs smell really good, dogs have 70 times the amount of those olfactory receptors than we do, that we don't have a very good sense of smell. On the contrary, humans have a pretty decent sense of smell. No, it is not nearly as good as the dogs, but it's not bad. Humans are particularly sensitive to those sort of chemicals generated by rotten or dead things, decomposition type molecules, because we don't want to eat something that is rotten or is breaking down. We want to eat something that's not decomposing because that would make us sick. So we're pretty good at smelling that. Certain other things we are not as good at smelling, but we are pretty good in that area. And your mom knows this because whenever you're like, Mom, can I eat this? When you pull it out of the refrigerator, she says, smell it. Does it smell good? And that's actually a pretty good rule of thumb that if it doesn't smell bad, it probably isn't bad. One of the interesting things about smell is that it's also very highly linked to memory. And if you start talking about a spring morning, you can probably kind of smell what a spring morning is like. Humans do have a pretty good memory for smells. It's closely associated with, how we remember things. Taste or gustation also uses chemoreceptors. These are different than the chemoreceptors in your nose. Those are found located in taste buds, and those buds themselves are protected by what are called papillae, or just a fleshy protrusion on your tongue. So when you look at your tongue, all those little bumps that you see, these are not taste buds. These are what are called papillae. Inside of a papillae, there is a taste bud. In fact, there's a lot of taste buds on each papillae. And so here's a great way of thinking of this, that each of those papillae is like an apartment building, and each apartment building has multiple windows. And inside of each one of those windows, there is a actual receptor site. So here is what a taste bud looks like. You've actually got multiple gustatory cells with little hairs that stick out, and then what's called a taste pore that lets a little bit of your saliva carrying that food into the pore where you can sample it. Now, this makes sense. We know that nerve cells don't regenerate real good. I mean, they don't regenerate. So if you damage those, they don't really heal. So we want to protect these really well. You don't want to have them exposed just on the surface of your tongue, because if you would burn your tongue, you could permanently damage some of these receptor cells. One of the interesting things right now in relation to smell and taste is that right now we're talking about the coronavirus, spreading through our population, and one of the characteristic symptoms is a loss of smell and taste. That virus affects the type of cells that line your upper respiratory system. As such, that can affect these cells in your nose, and smell and taste together, or olfaction and gustation, create what we call flavor. So if those are being affected by a virus or anything else, then you will have an impact on your sense of taste. When you look at your taste, you see a lot of diagrams that look like this. It shows a taste map of sweet, salty, sour, and bitter, but that's not the whole story there. There is a fifth taste, which is umami, which is a savory flavor. And you also even have water receptors in your pharynx or your throat. If you've ever drank water and you're like, wow, that's good water. Well, water really doesn't taste like anything, but you do have receptors that say that's water and that's good and I like it, because you need water to survive. When you look at this diagram of this tongue, this might give you that you can only taste these flavors right here. And what about this giant area in the middle? Can't you taste anything there? And the answer is yes, you can taste things in these areas. However, this is just a generalized map, and think of it more as like there's a higher concentration of bitter receptors here than in other places. There's a higher concentration of sweet receptors here than in other places. However, you still taste sweet in other locations, right? This doesn't even show the savory flavor or the umami flavor, which is another fifth type of distinct taste we can taste. All right, and so these chemoreceptors work very similar to the chemoreceptors found in your nose, in which molecules have to touch the right receptors. And if you get enough of the correct receptor stimulated, it then sends a signal that I'm tasting, a piece of watermelon or something like that. And then you are able to identify your food and say, that's good, and let's eat some more. Sight, your sense of sight or vision is, I guess, mediated by this organ, your eyeball. And one of the things that we always talk about in sight is how do you lose your sight? And the answer is there's a lot of different ways. The thing with light is not only do you have to physically gather the light, you have to accurately focus that light on a certain spot. That spot has to have the receptors to pick up the light, and then you have to process that image to get it a clear picture. A breakdown in any one of those things would cause you not to be able to see good. So first, let's look at the mechanical side of this. Right, your eye is covered with this clear covering called a cornea. Occasionally, the cornea can get scratched, or it can start to develop opaque spots in it, which are called cataracts. That would then restrict light passing through, and it would inhibit your vision. The hole that the light actually goes through is called your pupil. It's just that hole in the middle. The colored area surrounding that is the iris. The iris has muscles attached to it, which allow it to expand and contract in response to the amount of light you're getting. That's just to regulate, not having too much light on the back of your eyeball, which would hurt, and it would also damage things. Now, you want to focus that light that's passing through here with a lens. You actually have a gelatinous lens that helps to focus that light on one particular spot right here called the fovea. The fovea is the focal point for your light. If you are nearsighted or farsighted and you need corrective lenses, what's happened is that this lens is not putting that focal point exactly on that fovea. You may have known from using a microscope or a magnifying lens or something that you have to get it just right to have that focal point be just clear for you to see it. If your lens is not quite right or arranged right, you might not focus that exact focal point on the fovea. Corrective lenses help to readjust that so that way the light falls directly on this point in focus. That way you can get clear vision. The light that's being directed onto the fovea actually goes through a fluid inside of your eye called the vitreous body or vitreous humor. It's a fluid in there. You have a little bit of fluid here underneath your cornea called the aqueous humor. The term humor in anatomy means some sort of fluid. So that's got fluid in both cases. That clear fluid allows light to hit the back of your eye. Your eye itself is surrounded by this white part, which we call the white of the eye. The term for that is the sclera. The sclera is really, really tough. Human eyes are actually really rather strong. Not if they get poked by something sharp, but just from compression, they're actually rather strong. If you overpressurize the interior of your eye, which happens in a condition called glaucoma, that can distort the eyeball and the lens and cause you to start to lose your sight as well. That's another diagnostic thing for glaucoma. This pulled back part of your eye, even though the light's hitting the fovea, light is hitting other parts as well. This is coated with the retina, which is a collection of photoreceptors. So you've got the whole back of your eyeball, except for one spot where your optic nerve goes in and out and some blood vessels go in and out. This whole area is gathering an image, and then it takes that image and passes it along through the optic nerve to your brain. If you look at too bright of light, like you'd stare at the sun for too long, then you could damage those photoreceptors that make up the retina and you would lose your sense of vision, or welding or something like that. Now, the retina itself is made up of these photoreceptors called rods and cones. These two different populations of photoreceptors do really different things. Rods allow you to pick up low light images. This is what you use to see in the dark. Now, this doesn't let you see in color, and it doesn't let you see in super great detail, but it does allow you to see in low light. Cones, on the other hand, which they're just called that because the shape of the cell is a little bit different, the same with the rods, the cones, they're different shapes. They require bright light to see in, they allow you to see in color, and they allow you to see at a distance and see very clearly. Now, the interesting thing about this is that if you look at this diagram, this talks about receptor density and, let me wait for this to disappear. It talks about where the fovea here is in the middle. The peak cone density is right on the fovea, because this makes sense. When you look at something in light, you want to look directly at it, see it very clearly, and see it in color. That's what cones do. But we see that there's very few rods at all at the fovea. However, they're more on the periphery of your eye. Now, this actually makes sense. So this lens is going to focus right there on the fovea where you have your high concentration of cones. You see color, you see bright lights, you see very clearly, you see in a distance. At the same time, light is hitting this other part of your eye. And if you're trying to see in the dark, if you look directly at something, you actually can't see it good because that light's hitting the fovea with cones. But in the dark, if you look around, you can actually see pretty good in your peripheral vision. Try this sometime. When it's dark night, your eyes get used to something, stare at something, and you'll see that it's not clear. Look off to the side of it, and you'll see that you can see it more clearly. And that's because of the concentrations of where your cones and rods are. Now, one interesting thing about this is that your rods, which see dark, rely on a pigment called rhodopsin, and rhodopsin takes a while to generate. So whenever you expose your eyeball to light, you can't make much rhodopsin. Whenever you're not exposed to very bright light, you can start to generate rhodopsin, which actually explains if you've ever walked in from it's really bright outside, you walk inside of a room and you're like, wow, it's really dark in here. But then you give it 5 or 10 minutes and all of a sudden you're like, I can see just fine in here. What's happening is you're utilizing more of that rhodopsin to be able to see. And if you have been walking around in the dark for quite a while, and then like some light shines on your eyes or something, and then all of a sudden after that you can't see for a while. You just lost your night vision. That is, this pigment, if you get somebody shines a flashlight in your eyes, you lose some of your rhodopsin. Okay, it causes it to break down, and then you can't see very good in the dark for a while until you regenerate it. Kind of, explain some things. One of the other interesting things about cones is that they come in 3 different colors, like red, blue, and green. For someone who is colorblind, they do not have enough of a certain population of cone. Typically, it's the red-green differentiation. So typical color blindness, people would be unable to distinguish between the color red and the color green. They would perceive it as the exact same color because they don't have the cone population to differentiate between the two. You can have extreme cases of color blindness where people see somewhat monochromatically, where they just see in shades of black and white or gray. But those are rather extreme cases of color blindness. One of the interesting things about your whole sense of sight is it takes a lot of visual processing to make you be able to see stuff, because really you're taking some information from your photoreceptors, and then your brain has to coordinate 2 eyes seeing the same thing, overlay those images, and then allow you to calculate distances and whether something's moving towards you, like if you try to catch something. So there's a huge amount of optical processing that goes into you seeing something. Because of this, your brain really fills in a lot of blanks for you, and that allows us to trick your brain using optical illusions. So looking at this optical illusion I have, if you stare at one part of it, you tend to think either the circles are moving or it's a spiral, and it's really not. Looking at this one, there's 2 different images in here that people see. There's some people see an old woman. Here's her nose and her mouth and her eye, and she's looking towards me. And some people see a young woman looking away. There's her eyelash and her nose, and this is her jawline and her ear, and she's looking into the distance over there. So you can interpret that either way, but your brain works really hard to recognize and see faces. We're wired literally to see and recognize faces because we're humans and we look at each other's faces. That's why people see faces in the clouds and on trees and everything else. These gray lines are not sloped. They are horizontal, even though when you look at it, our brain tends to want to interpret these different colored bands as making it look like some of these lines are sloped. This one's just odd. It's using a 2-dimensional image to make us think that there's 3 dimensions here, and it just doesn't match up. Spend some time looking at this one. Here's a picture by the artist M.C. Escher. This was done in 1953, but he did a lot with perspective, and we start to make sense of an image, and then all of a sudden it changes its perspective, and then it allows you to make these really interesting drawings that he liked to make. What color is the boy with the red hair shorts? And the answer is we don't know. The boy with the red hair is buried. There's another boy here who does have on green shorts. Because we're so focused on associating faces, we want to associate this face with this body. We're kind of wired to do so. Hopefully that's not really that boy's head. This is another example of kind of a perspective change where there's really no good answer. How many legs does this elephant have? It has several. Spend some time looking at that. And then finally, there's this image. Whenever you look at it or stare directly at it, it looks like they're rotating. They're really not rotating, but it does give that perceived image of that it is, because you're really trying hard to process that visually. Kind of wild. Okay, as far as your senses of equilibrium and hearing, in both cases, the sensors for that, or the receptor cells, are found in the inner part of your ear. They're also both types of hair cells, which are a simple mechanoreceptor that has a cilia or hair sticking out of the cell. And whenever that hair gets moved, it then causes that cell to send a signal. Now, because they're close together, even though they're not exactly the same thing and they're not the same organs, we'll look at where they're located. They are close to one another. And what can happen is if someone gets a really bad ear infection, not only will it affect their hearing, it can also affect their balance. So some of you may have before gotten a bad ear infection, and as a result, you may have lost your sense of equilibrium or been able to balance very good. Right, so the ear itself. Your ear is divided into the middle, inner, and external ear. The external ear is the part outside of your body that you can see right here. You've got this auricle, or the outer part of the ear, that helps to channel sound into your ear canal, or external ear canal. The external ear canal has ceruminous glands, or wax glands, that secrete wax here. Wax in your ear is not there to be gross. It is there to, it does have antibiotic properties to keep bacteria from growing in your ear. It's there to trap materials, so that way those materials don't end up stuck onto the eardrum or the tympanum right there. That's the purpose of wax, right? Your external ear terminates at your tympanum, or your eardrum. So if any of you are familiar with band, the timpani are the really big drums. So tympanic means drum. So that's your tympanic membrane. That is what your eardrum consists of. So whenever you hear sound, the sound vibrates your eardrum, and then that's going to transmit that sound on, right? That gets you from the external ear into the middle part of the ear. The middle part of the ear, through here, has 3 small bones in it: the malleus, incus, and stapes, or the anvil, hammer, and stirrup. They're the smallest bones in your body. There's 3 in each ear. And these bones, actually, the hammer is connected to the eardrum. And so when the eardrum shakes, it shakes these bones, and those then transmit the vibrations through those bones to the actual part where you actually really pick up the sounds neurologically. So far, this is all mechanical transmission of sound, which actually explains if you have an ear infection, that ear infection is in the middle part of your ear, right here. If those have fluid around them, they don't vibrate as good. And if they don't vibrate as good, you might have had a head cold or something, and then it was hard to hear, or you couldn't hear clearly. That's because these bones aren't vibrating. Your middle part of your ear actually drains down to the back of your throat through this tube called the eustachian or auditory tube, and that allows fluid to drain out of this. Now, occasionally, if you get an ear infection, this gets clogged and you can't drain the pressure, and it makes your ear hurt. If you get a bad enough ear infection, it can overpressurize and rupture or tear your eardrum. For someone who has tubes in their ear, they make a small incision in the tympanic membrane and put a little port in it to allow air into the middle ear. That helps this drain down the auditory eustachian tube to the back of the throat to help prevent more infections in your middle ear. The actual hearing part, where you actually receive the signals, is in the cochlea, which is this part right here. It looks like a coil or snail. That is where you actually have your receptor cells. Your organs of balance are right here, and these semicircular canals up here. They're close. They're not exactly the same thing. Let's see. On the inner ear, you have vibrations that enter the vestibule, and then those sound vibrations travel down the cochlea, where they're received by hair cells. So it enters to the vestibule or the opening, and then those vibrations travel down this coiled structure. Depending on where in that coiled structure or cochlea that those hair cells get stimulated, it tells you the pitch of it. The rapidity tells you the frequency, and how much they get perturbed tells you how loud the sound is. So here are these hair cells. I have a good picture. There are the hair cells that are actually in your ear. Okay, as those get shook by having a vibration pass over them, you then interpret that as a sound. Now, here's an interesting picture showing hearing loss. So these are the nerves lining right here, these hair cells. You can see the hair cells right there, which are these right here. Okay, those hair cells line that cochlea, that coiled part. And so as sound is coming in, depending upon the wavelength of the sound, it will stimulate different areas of those hair cells and tell you different pitches. How much they move will tell you the magnitude of the sound. So what has happened here is this person has experienced loss of hair cells between here and there. That means that they cannot hear certain pitches. They might be able to hear high-pitched things and low-pitched things, but they might not be able to hear very good with just mid-range sounds. They just don't have those hair cells. They cannot physically, these aren't around to vibrate, so they cannot hear within this range. For people that are around like loud droning things, like if you run heavy equipment without ear protection for long enough, you can damage a certain range of these cells and not be able to hear very good in that particular sound range. Children are because their cochlea are smaller, they're actually able to hear higher pitch sounds than you can as an adult. You may be familiar, they have ringtones that adults can't hear because it's too high pitched, yet kids can maybe hear that. Keep in mind, kind of like with the eye, like anywhere along this whole process of seeing or hearing that either that light wave or that sound wave or the receptors to pick up the light wave or the sound wave get damaged, then you have a breakdown in this. So in terms of hearing, you can have hearing damage come as a result of these inner ear bones or middle ear bones being damaged. Repetitive ear infections can damage those bones. You wouldn't be able to hear then. Sometimes you could have damage on these cochlea and you wouldn't be able to hear then either. If you rupture your eardrum and your eardrum is scarring on it or it thickens, then once again, it doesn't vibrate as good and then you can't hear as well. So a normal hearing aid just simply picks up external vibrations and magnifies them to make things vibrate a little more so you can pick it up. In the case of this particular type of hearing loss where you lost those hair cells, it doesn't matter how much you magnify the sound, permanently, you will not be able to hear in that range. There are types of implants called cochlear implants that directly stimulate this that can allow people to hear that otherwise wouldn't be able to. As far as your sense of equilibrium, I think maybe I skipped it. Your sense of equilibrium, let me quit flipping here so bad, is carried out by these things called ampullas and these hair cells within these loops within your inner ear. These are called your semicircular canals. Now, this is very interesting. There's two different types of receptors here. Okay, one are what are called ampulla. These are hair cells that have a big glob on top. And as you move back and forth, just like a little wobbly bobblehead thing, this bobbles back and forth. That tells you which way you're moving. Like if you're accelerating, you're tilting one way or another, that allows you to determine whether you're moving and which way. You also have freestanding hair cells that aren't coated by anything that are in a chamber with little pieces of literally ear rock called otoliths. That's literally what that means. Your body actually makes small mineral crystals that slide around inside of your ear. And as they slide around, they brush against these hair cells and stimulate them. And that tells you if you're going one way or another, if you're upside down, you're able to tell how you are oriented somewhere. So if this gets an infection in it, like an ear infection, these aren't able to operate either, and then you can, that will impact your sense of balance. Okay, we'll go ahead and stop there.